These are the highest levels of the brain. We're to the most sophisticated integration functions we'll talk about in lecture. And this large crease here is the longitudinal fissure between the left and right cerebral hemispheres. This large structure on the dorsal side of the brain stem is called cerebellum. And that's very important for muscular coordination and for carrying out any fine motor acts, like in craftsmanship, athletics, any kind of complex movements that works this big hemisphere on the back of the brain stem.
And what are they called?
Cerebellum.
That's a pretty name.
And the meninges, well, none of these have evidence of the duramater on them. If you see that, it's going to be very tough outer layer. Let's see. Again, I'm finding none of the things I need here, so I'll grab another brain. Find one that has it. These were better off last week than this week. They're getting picked over. So this brain is covered by the outermost meninge, which is called the duramater or tough mother. And the whole brain and spinal cord, the whole central nervous system covered by these three meninges. The middle one has all these spider-like appearing things on it. So it's called the arachnoid meninge. And it goes over all these grooves and without going deep inside. So it's like taking saran wrap or plastic wrap and wrapping it around the surface. It doesn't go into all the little depressions. And I just remembered I need one more thing from over here. Getting all my acts together is not easy.
I was even recording, so I got a photo, and I was.
So I need the cookie.
At least you notice now instead of like at the
I know.
end.
This cookie is pretty good. So you can barely see the arachnoid meninge. It's starting to dry up here, and you see it between the creases. I could probably get a pin underneath that, but that would be the only other one I could possibly test on. But you need to know them for lecture. The pia mater is invisible. I mean, it's too thin to see. It follows all these grooves, goes down into those crevices.
So you want tests on that?
No, you do that for lecture. So this model shows you what the gyri and the sulci are. Sulcus, we already learned, is like a valley, right? It's an indentation. And the gyrus is like a hill. So if you think about a little stick man gyrating on the hills and sulking in the valleys, that's how you remember that. Now the brain, the cerebral hemispheres and the cerebellum have lots of gyri and sulci. Some of the sulci are very complicated. They go inward and then off to the side. And what the sulci do, the gyri and the sulci, is they add surface area to the brain. So you have more gray matter.
Can you point out the difference between the gyri and the sulca again?
Gyri is a hill, sulci is a valley. So all these indentations are sulci. All these hills are gyri, gyrations. So the little man gyrating on the
Is
hills.
that like a horizontal cut of the brain?
Yes,
Wait, how would
to
you
make cookies.
how would you identify that? Like, in the
Well, you just have to know what they are. It's pretty hard to point it out, but
So we'll
if I would have to have each of these questions will have a sentence.
describing it.
Yeah, what to look for. Name these undulations, these hill-like undulations. Name these deep crevices. So the more gray matter you have, the more integration your brain can do, the smarter you are, right? So gray matter represents nerve cell bodies and small or short unmyelinated nerve fibers. That's the basic color of nerves. But nerve fibers that convey nerve impulses over long distances have to be insulated with myelin to speed up the rate of transmission, and myelin makes them look white. So white matter represent myelinated nerve tracts, and gray matter represents integration centers where decisions are being made. So that's the difference between gray matter and white matter. Okay, looking at the surface of the brain from underneath, this illustration, we can see a series of objects, structures, as we go from the anterior to the cranial to the caudal end of the brain. So the first thing we see are a pair of structures that look kind of like antennae, these flap-like structures. These are fresher here, but they're getting kind of messed up. There they are there and there. Hopefully we'll have a nice one for the exam. Here they are too. Those are the olfactory bulbs. And olfactory nerves from these bulbs go through the cribriform plate of your ethmoid into the nasal cavity. And that's where you sample the air, and receptors respond with impulses to the brain that are converted into a sense of smell. So they're associated with olfaction. About a third of the way back, we're going to find these optic nerves come in from each eyeball, and they cross. And the crossing over from one side to the next, which is seen better in this illustration than we have on the real brain. We'll have to do some dissection to show it. Well, there's part of the cross right there. What you can put down on your notes is X marks the spot. That's called the optic chiasma. Now what that does is each eyeball is getting a separate image slightly apart, and sending that image to the brain, and then sending collateral branches to both hemispheres. And they're going to integrate that information and come up with a virtual picture of the world that's three-dimensional. You're not really seeing the real world, by the way. Your eyes' receptors are converting information into nerve impulses. The nerve impulses are going into your brain, and they're being processed many times. And then what you see is a virtual image of the real world created inside your brain. You're not seeing the real thing. It's like a camera takes an image and sends it through a electric cable into a device that then converts that into a picture. You're not sending pictures through the wires, you're sending electrical information. Same with a microphone, picks up vibrations of the air, turns them into electrical signals, and then an electrical instrument turns it into an amplifier and all that stuff, turns it into sound that's produced on a speaker. So nerve impulses are all that's going on in and out of the brain, not pictures or sound. Now, in the middle of the base of the brain, we're going to find a rounded object. To me, it looks like a chickpea or a garbanzo bean. It's a different name for the same pea. And that is your pituitary gland. Now, the pituitary gland rests in the sella tursicum right there. It's not Ross the cowboy that's in the seat, the saddle. It's your pituitary gland.
Can you point it out?
There's the pituitary right there. Yeah. There it is right there. Okay, so you can see that right in the middle of the base of the brain. Whenever we cut these brains open, though, the pituitary falls off. So we need a nice whole one here for that. Then just below that, we're going to find two rounded mounds of tissue that we call the pons. They're not very prominent in a sheep brain, but much more so on a human brain. See how that large anterior protuberance is? That's the pons, and it's part of the brainstem. Here is the medulla oblongata, the lower part here, before it connects to the spinal cord. Medulla oblongata, pons, and then this little section in here is called the midbrain. We'll be talking about that more in lecture. So those are the basic items you need to learn from the external view of the brain. Now, when we open it up, and this one's a particularly good one, I think, what can we see? Well, the most prominent feature is this big white band that loops around here. And you see it there also. And here you see it in cross-section. Now, what that is are bundles of white myelinated fibers, nerve fibers, that communicate between the right, they allow the right and left cerebral hemispheres to communicate with one another. It's called the corpus callosum. To me, it looks like a rubber band, or calamari that's been sliced up for the Rhode Island, official Rhode Island appetizer. Anyway, so that's pretty easy to see. Right at the base of the posterior part of the corpus callosum is a little gland called the pineal gland. It always falls out of the cross sections of the brain, so I don't think I can test it on that. But it's on the model there. And that gland produces melatonin that's associated with your sleep and wake cycle. Some people take melatonin too. Yeah, have some melatonin. That's terrible. Some people have melatonin to help them sleep or to catch up after flying a long distance in a fast jet to deal with jet lag. Okay, some other features we can see on the model or on the sheep brain are some of the, you can see here better the ventricles. This model, the cookie I call it, has the right and left ventricles in the cerebral hemispheres. Don't ask me which one is 1 and 2. I don't know if it's right or left. But those are the lateral ventricles. And they're filled with cerebral spinal fluid. The fluid is created by capillary filtration from networks of capillaries called choroid plexus, or plexuses plural. So there are 4 ventricles. The 2 lateral ones you can only see in this model. They flow into the 3rd ventricle, which is a very shallow one. You can't see it on the sheep brain because it's too thin. On this model, it's just in here around the inside of the thalamus. And then there's a little aqueduct, cerebral aqueduct, that carries the fluid to the 4th ventricle, which is right at the base of the cerebellum. From there, the fluid goes down the central canal, the spinal cord, and then around between the arachnoid and villi and the pia mater. And then up here, it's reabsorbed by arachnoid villi, which you'll learn about in lecture. Now, the choroid plexuses can be seen at least in the 3rd and 4th ventricle right here. That's where the cerebral spinal fluid is formed in each of the ventricles. It's a filtrate of blood. Because of the presence of astrocytes, those blood vessels are tighter than the systemic blood capillaries and blood vessels. So what those capillaries are tighter. So this forms a blood-brain barrier that protects the brain from potentially harmful substances in the blood. Now, the cerebellum has white matter and gray matter, but the white matter is in the shape of a tree, in fact, a tree of life. So it's called the arbor vitae, tree of life. And the leaves, the gray matter, of course, where the cells that do the integrative functions are located. So the cerebellum sits right on the back of the brain stem, and you see lots of white matter here. Sensory information coming from your joints and your muscles comes up through the brain stem and then is into the thalamus. This midsection of the brain is then it's projected onto the sensory cortex. We'll talk about that in lecture. And then the motor cortex sends out signals to control your muscular actions. And collateral branches of the sensory input and motor output are both sent to this structure, the cerebellum. And it integrates that information and it modifies the signal coming out of the motors area of your cerebral hemisphere so that it adjusts it, makes fine adjustments so that you can produce fine, skilled movements. So anything you do that requires practice, You get better and better at it the more you practice by the interaction of this part of the brain with the cerebral hemispheres, because it monitors both motor output and sensory input from the joints and muscles. So and it also receives input from the inner air apparatus as far as equilibrium is concerned. Your position relative to gravity, whether you're accelerating, decelerating, all that information has to be integrated here. And without the interplay between these two parts of the brain, you could never perform the kinds of skilled actions that we do in arts and crafts and music and athletics and all kinds of fields, even studying for exams. Okay, so that's the arborvitae. This is the cerebral aqueduct I mentioned. I can test on that. Did I leave anybody out? There's a picture of what the pineal gland would look like in a sheep brain before it falls out. So we'll have to use this model. Unfortunately, models have numbers and colors and stuff like that, rather artificial, but we can do that. So this model could be used for parts of the brain, parts of the digestive and respiratory system. And then we also have this other model we just got recently, which was built by a 3D machine, I think it was. And here are your cerebellums. They go on the back there, and then there's two hemispheres. Their gyri and sulci are much narrower and sort of in rows. It reminds me of an Italian pastry that's made once a year. They look like a big quahog and it's stuffed with custard. And so this model, I can see the brain stem, I can see the pons. These, I'm not sure what are. I don't know, there's a key to this thing that's useful, I suppose. But we'll probably use the real thing more than anything else for this exam. I don't even want to put it back together, so. But this was sent to us, thought we'd be interested and ordered more. I don't have the patience for it.
So you said that this was a cerebellum?
Yeah, there's
And
two.
where's the arborvitae?
Well, they didn't cut it open, so.
Oh, so it's inside? You can.
I don't think these come apart, but yeah, they would be inside, but
What about-
certainly you can see right there. See that tree of life, the branching tree?
Yeah.
Yeah, that's arborvitae.
Okay.
And then, well, you know about Arbor Day, when you plant trees, and that's, arbor means tree.
Yeah.
So, put this over here again.
Oh, it does come apart.
Oh, it does come apart. So there's your arborvitae. So it does come apart.
Okay, so that's
But it looks
inside.
more like Christmas tree than the tree of life. They made it into an evergreen instead of a deciduous tree. Okay, so you figure that out. Okay. I don't know. I'm not a fan of models. So, I think we've covered everybody. And so there will be, I can put models out on the exam. I can put sheep brains. And we'll probably have a couple of sheep brains, we'll have a couple of sheep hearts, we'll have the models of the heart. And these other models, human models around here, and they will be testable. So I'm gonna put these, well, I'll leave them out for you to study first. And then we have to do reproductive system. And that's gonna be a struggle because we have to find a male and a female. What about Big Mama? Think Big Mama is? Anybody remember what Big Mama is? No? It's in this group, right?
Yeah.
The black one, right?
It's still here? Yeah, go get Big Mama, bring her down.